Hello everyone, welcome back for the last video in Chapter 3 of Human Physiology. So this video we are going to talk a little bit about how proteins are made inside the cell. Most of that is on your own work, but we're majorly going to kind of focus our attention on, with protein synthesis, how does this sometimes go wrong and how can that affect how physiological processes are supposed to go. So in your on your own work, you should become familiarized with something called the central dogma of molecular biology. Basically, this covers all the necessary steps to tell you how is a protein going to be made. This is not only going to involve how DNA strands are replicated, but also how messenger RNA molecules are made through the transcription process by an enzyme called RNA polymerase. And then those messenger RNA molecules are then exported out of the nucleus where they are captured by a ribosome, and then through the translation process and by using the genetic code, which we will talk about a little bit later, the ribosome reads the messenger RNA sequence and is able to synthesize a protein in which each amino acid that is incorporated into the protein is determined by a stretch of 3 nucleotides that are found in the messenger RNA sequence. So those stretches of 3 nucleotides that I talked about in the mRNA, each one of those is called a codon, and every codon will code for one of the 20 amino acids to be incorporated into the protein in a specific predetermined order. Now, the genetic code, which you can see in this complicated looking table here, can seem a little bit intimidating, but just know I am not requiring you to memorize this or even remember a whole lot about it, outside of a few things. Now, first, before I get to that, let's look at what we have over here on the right. Let's say that we have a DNA molecule that goes from 5' to 3' in order: ATA, TAC, AAA, GGC, CGT, ATC. By using the transcription process, the messenger RNA molecule is going to read UAU because this is complementary to what is found in the DNA, and it has to be UAU instead of TAT because you'll remember that thymine is not found in RNA molecules. So from ATA we go UAU, AUG, UUU, CCG, GCA, and then UAG. So that would be the sequence of the messenger RNA made off of this DNA template. Now, if we're going to make a protein, we need to look at each of these codons and find them in the genetic code so that we can know which codon, in this case we've got a UAU codon, which amino acid does that correspond to. Well, for UAU, that is going to correspond to, if we go first letter is U, second letter is A, and then third letter is U, that should correspond to the amino acid tyrosine. But here's the problem. We can't just immediately launch in and say we're going to put a tyrosine here, because in order for a protein to be built, you have to start with what is called the start codon. Not every single part of the messenger RNA is necessarily going to code for the protein. So the first thing that the ribosome is going to do is it is going to scan along the messenger RNA molecule from 5' to 3' until it encounters the first start codon, which is AUG. So the first UAU does not really mean anything. We don't start building the protein until we get to AUG here, which of course codes for the amino acid methionine. Because methionine is always associated with the start codon, most of our proteins are always going to start with methionine, unless after the protein is made, that methionine is chopped off, which can sometimes happen, but methionine is always associated with the start codon. Okay, and then after that we go UUU, which corresponds to phenylalanine right here. Then CCG, which corresponds to CCG, that is proline. And then finally GCA, which is alanine. And then finally we get to UAG. if we find what UAG stands for, it does not actually correspond to any amino acid. It is actually one of the 3 stop codons: UAG, UAA, and UGA. If you're having a hard time remembering what those are, a funny little mnemonic device I learned recently is that UAG stands for you are gross, UAA stands for you are annoying, and UGA stands for you go away. So if you just think of someone you really don't like, you should be able to think those up pretty quickly, which I kind of find amusing. Okay, so those are our 3 stop codons. So when the open reading frame here, which starts with methionine, which starts with AUG, once we hit a stop codon, we are going to stop making the protein. So this protein would literally only contain 4 amino acids: methionine, then phenylalanine, then proline, and then alanine. Okay, so once we have our messenger RNA molecule, to go into a little more detail on how this whole process works, once you have transcribed the mRNA off of the DNA template, and keep in mind, we're only going to make 1 messenger RNA for every gene, so 1 gene on the DNA provides the instructions for making 1 protein. So therefore, 1 mRNA transcript that comes from that gene, that mRNA sequence is going to provide the instructions on making the same protein, and those instructions should be exactly the same. So once that mRNA molecule is exported out of the nucleus, it gets captured by a ribosome, and then the ribosome is going to use its enzymatic activity, which of course comes from the ribosomal RNA, which is unusual for us because usually we associate enzymatic activity with proteins instead of RNA. The enzyme activity of the ribosome is going to use dehydration synthesis to form peptide bonds between amino acids that are attached to transfer RNAs. The transfer RNAs bring each corresponding amino acid in so that we know that this codon corresponds to this amino acid. So the first tRNA that will come in will bring a methionine, and then a phenylalanine, and then so on and so forth. So the enzyme activity of the ribosome will, once we get those amino acids close together, because the tRNAs are close together, we will forge a peptide bond between them, and then we just keep bringing in new tRNA molecules so we can keep adding amino acids to the growing polypeptide chain by dehydration synthesis. And the protein is going to contain as many amino acids as it takes for us to eventually reach a stop codon in the mRNA transcript. So you may either have a very short protein or you may have a very, very long protein. It just depends on how many codons are in between the start codon and the stop codon. Okay, so ultimately, when it comes to knowing the specifics of how proteins are made, why do we care? Why do we even care? So why do we need to know that to know how things like how the heart beats, how the brain functions and sends electrical signals? Why do we need to know how proteins are made? Well, I've said this before, but basically every physiological process is mediated by proteins. So some proteins that we have already talked about before would include the sodium-potassium pump. That's a protein, and a very, very important protein at that. Insulin, the hormone, is a protein, and so is the insulin receptor, which we'll be talking about in the coming chapters. The CFTR chloride channel that becomes mutated in cystic fibrosis, of course, is a protein. And then collagen and keratin, which are important parts of the extracellular matrix and of connective tissue, they are proteins as well. We care about protein synthesis because in every physiological process, it's proteins that are doing all of the heavy lifting. Therefore, if our cells are not doing a good job of making proteins correctly, the physiological process itself is not going to work right, and that is how you get a lot of diseases. So how can protein synthesis get screwed up? One way to do it is to accumulate mutations in either the DNA or the RNA. A mutation is literally just a change in a nucleotide sequence in either the DNA or the RNA. So if we change the order of nucleotides, we can potentially change which amino acids get incorporated into the protein. And if we do that, the protein potentially can fold a lot differently and then either lose function or take on a bad function. So as far as point mutations go, so a point mutation is just a single nucleotide change in the DNA. Point mutations come in a variety of different forms. Before we talk about them, let's look at what the no mutation aspect is here so we can see what things are supposed to normally be like. Let's say that we have DNA that is TTC. So the mRNA would then be AAG because that is complementary to what you see right there. If you look up AAG on the genetic code, you are going to get the amino acid lysine. Lysine is a basic amino acid. It is hydrophilic. It is positively charged. We talked back in chapter 2 about what that specifically means. So let's talk about the different kinds of point mutations. First, you have what are called silent mutations. Let's say that the DNA here gets changed from TTC to TTT. So we change one nucleotide, we change C to T. And therefore, in the mRNA, we go from AAG to AAA. But notice what happens here. Even though we change the nucleotide sequence, we still end up with the same amino acid that's incorporated into the protein. The reason why this is called a silent mutation is because even though the DNA and the RNA change, the protein itself is completely unaffected because we have not changed which amino acid actually gets incorporated into the protein. Therefore, the protein will fold normally and it should function normally. A silent mutation should have absolutely no impact on cellular function, protein function, etc. Therefore, we call it silent because we don't notice it. A very bad mutation that is going to very drastically impact protein function and physiological function is what is called a nonsense mutation. In this case, let's say if we normally have TTC, let's say we change that first thymine to an adenine. So now we have ATC. At the mRNA level, we now have UAG, which of course is one of our stop codons. So if that's the case, instead of incorporating lysine into the protein, we just stop making the protein right here. Maybe we were supposed to incorporate another couple hundred amino acids after that lysine, but here it doesn't matter. This is going to be where we stop making the protein. So what we're probably going to do here is either make a protein that is way too short, or we're going to make a protein that is so short that the cell isn't going to like it and the cell is going to destroy it, which is what happens in cystic fibrosis. Nonsense mutations are very, very bad, and we want to avoid them. The final kind of mutation is called a missense mutation. This is the type of mutation that actually does change the identity of which amino acid is incorporated into a protein. Let's look at what is called a conservative mutation. In this case, we end up incorporating the amino acid arginine. The reason why this is called a conservative mutation is because we have gone from lysine, which is a basic amino acid, to arginine, which is also a basic amino acid. Even though these are different amino acids, they both fall into the same class of amino acids, and they are going to function very, very similarly. So because of that, conservative missense mutations generally are going to have minimal effects on the protein function and how the protein folds. There may be some effects, but we're going to minimize the potential bad effects here by making a conservative mutation. Now, a non-conservative mutation would change the identity of the amino acid to something that falls in a completely different class. In this case, we go from lysine, which is basic, to threonine, which is polar and non-charged. Now, they're both hydrophilic, so this isn't as bad as it could have been, but threonine and lysine are going to behave quite a bit differently because they fall in different classes. We could definitely end up with a very bad non-conservative missense mutation if we swap out lysine for something that is hydrophobic, like alanine or isoleucine. So here's some examples of diseases that are actually caused by either mutations or problems with the protein synthesis process. Number 1, sickle cell anemia. So this is caused by a non-conservative point mutation in the beta-globin gene, which is one of the genes that are used to make hemoglobin. A glutamate amino acid, which is hydrophilic and acidic, is swapped out for a valine, which is hydrophobic, and that causes the protein to not get made correctly. It becomes a more hydrophobic protein, it doesn't fold correctly, and it impacts red blood cell physiology. And we will talk more about that in Chapter 20. Cystic fibrosis we've already talked about, but we can get a little bit more context here. This is a nonsense mutation in the CFTR gene. The ribosome hits a stop codon prematurely, and the ribosome releases the protein before it can be properly made. This protein does not fold correctly, and it gets destroyed by the lysosome. So this is the worst case scenario. The protein just is totally non-functional, and it is not able to do its job. A variety of different cancers are often caused by DNA mutations, and these mutations usually come in genes called either proto-oncogenes or tumor suppressor genes. If it is a proto-oncogene, the proto-oncogene is usually going to be a protein that acts to encourage progression through the cell cycle. So if a proto-oncogene gets mutated, usually this can cause cancer if the mutation causes the protein to become especially active, more active than it normally would be, and we call those gain-of-function mutations. Tumor suppressor genes, the protein products of these tumor suppressor genes, inhibit progression through the cell cycle. So if a mutation causes a tumor suppressor gene to lose its function, then cells can go through the cell cycle unregulated and can cause a lot of different cancers. All right, that is going to do it for this final video in Chapter 3. Thank you for your attention, and I will see you next